All right, it's my pleasure to introduce Sujita. She is originally from Nepal. She has a bachelor's degree in fisheries science and a master's degree in aquaculture. Currently, she's working on her PhD with Dr. Matt Griffin at Mississippi State University's College of Veterinary Medicine. Her project involves the development of vaccines against bacterial disease in catfish aquaculture. In her free time, she likes reading books and taking long walks, and has recently developed an interest in sketching and painting. It's great to have some work-life balance. Take it away, Sujita.
Thank you, Dr. Fells, for the nice introduction. So yeah, I'm Sujita, and today for the AFS-FHS Summer Seminar Series, I'll be presenting a topic, rifampin-resistant Idursia piscicida as potential live attenuated vaccine candidates in channel and blue catfish hybrids. And I would like to thank AFS-FHS for this opportunity to present. So talking about the U.S. catfish industry, it is the largest freshwater aquaculture industry in the U.S. with predominant culture of channel catfish, Ictalurus punctatus, and hybrid catfish, which is a cross of female channel and male blue catfish, Ictalurus furcatus. So the U.S. catfish industry had a sale of $437 million in 2023 for the food size fish. The largest producer of catfish is the Mississippi State and is followed by Alabama and Arkansas. But as the industry shifted towards the intensification, there has been a threat of emerging diseases in the industry. Bacterial pathogens are one of the most important pathogens that is causing this loss. So why is emergence of Idursia piscicida important? So as Dr. Nurul Huda told, so the hybrids have been developed to have superior growth performance, feed conversion ratios, as well as tolerance to low dissolved oxygen. And also, addition to these traits, they are also resistant to several of the channel catfish pathogens. And the industry has now shifted from channel to hybrids being more than 50% of the cultured species. But as you can see in this report developed by Aquatic Research and Diagnostic Laboratory, Stoneville, Mississippi, that from the past 10 years, hybrids have more cases of Idursia piscicida, and more than 90% of the cases of Idursia piscicida is diagnosed in hybrids. So what is the impact of these bacteria? Associated with mortality and morbidity, it also have problems with reduced growth because of the restricted feeding practices that is practiced to prevent the spread of infection. This disease also increase the feed costs associated with the medicated feed. And as Divya talked about the antimicrobial resistant, using the medicated feeds may have a chance of more antibiotic resistance in the industry. So combined all these affect the profitability of the farm. So there is a need for a better strategy to prevent and control the disease, which demands industry-defined need for an effective vaccine. So rifampin resistant rifampin resistant have been used to create live attenuated vaccines from the past, and it has been successful so far. What does rifampin do is it inhibits the bacterial DNA-dependent RNA polymerase activity, and the resistant bacteria are known to carry spontaneous mutations in various parts in the RPOB gene, which produces changes in global protein expression profiles or altered lipopolysaccharide biosynthesis, rendering the bacteria less virulent or avirulent to cause disease. One of the important or successful example of rifampin mutant as a vaccine in aquaculture is the ESC live attenuated vaccine developed at the Mississippi State University, which is orally delivered using the oral vaccine delivery system, as you can see in the picture. So as Idursia ictaluri and Idursia piscicida belong to the same genus and are closely related, we hypothesize that creating a vaccine the same way may provide protection against Idrosiella piscicida. So keeping this in mind, this project was planned to create a potential vaccine against Idrosiella piscicida in channel catfish with the objectives to create rifampin mutants from various isolates of E. piscicida representing known variants, and to test the attenuation of the created rifampin mutants as a potential vaccine candidate in hybrid catfish. Now, selecting the isolates of these representative groups of the Idrosiella piscicida have been an important part of this project because Idrosiella piscicida have 5 genetic variants or phyletic clades that had been identified in the previous study in 2021. And we doubt that a vaccine from one clade may not work for the another. To overcome this problem, we selected representative isolates from all clades to create the rifampin mutants and test them for cross-protectivity between the groups. Next step for the development of the rifampin mutants, we made media with different concentration of rifampin from 0 to 360 microgram per mil, and then selected isolates were initially passed at 5 microgram per mil of rifampin. Then the isolate growing in the highest concentration was grown again. 3 separate colonies were expanded in the PHI broth, and successive passages of 5, 10, 15, and 20 microgram per mil was made with each step. Cryostocks were made at each step of this process so that if we can revisit the isolate one more time and continue the passes if there is any problem in between. All the isolates were passed until the final concentration of 360 microgram per mil, and each isolate took variable number of passages to reach 360 microgram per mil. We did passages of 4 to 45 in each step. And we created a total of 19 mutants from this research. Now for testing the attenuation of those developed new mutants, we did 2 experiments. For the first experiment, we used the flow-through tanks with 5 tanks per treatment and 20 fish each tank. We acclimatized the fish for 1 week and then injected them intracellularly with wild type and their counterpart mutants, RIF mutants, with the dose of 1 times into the 4 CFU per gram of fish, and the sham fish were injected with PBS. We observed the fish for mortality and morbidity for 14 days and collected the dead and moribund fish. We recovered the bacteria from the kidney, and the colony PCR was done in real time to confirm the infection. After 35 days, the fish were challenged with the virulent MLSA1 S11285 E. piscicida through intracellular route to check for the protection. At the dose of 1 times into 6 CFU per fish, and negative control fish were given PBS, and positive control fish were injected with the same dose of bacterial injection. So these are the 10 isolates that are used in this study from 3 different MLSE groups. Now looking at the results, we did not see the expected amount of mortality that we targeted. We targeted LD50, but we only got LD20. We were lower on the immunizing dose, or for mutants, maybe we have over attenuated the mutants, so we did not see the level of mortality that we expected. However, based on this analysis, the wild type and the mutants mortality when compared side by side, we can see that 3 isolates out of 10 attenuated evident by the blue bars in the picture. While low levels of mortality were observed in the wild type, there was a negligible mortality in the mutants, indicating that there is some level of reduction of mortality. So the re-challenge study was done. Now looking at the protection evaluation, we can see that the wild types provided significant protection against the S11285 compared to the control, meaning that Idrosiella piscicida is a good candidate for a vaccine. And also this challenge showed that the group from, the MLSA group, different MLSA groups can provide protection against a different MLSA group, because these were from different groups and we challenged it with the same MLSA group 1, and we see significant protection, meaning that Idwasella piscicida may have cross protection between the MLSA groups. Now, looking at the results from the mutants, out of 10 mutants, we saw that 4 of them were protective against the S11-285 compared to the sham. But as we can see that we did not see high mortalities in the attenuation test, and also 3 of them were attenuated and 4 of them saw protection. So we need to revisit this and redo the experiment one more time. Now, going on to the second experiment, we use the same experimental design as the first one and check the attenuation for other 7 mutants. And we use higher dose this time of 3 to 6 times into 5 CFU per gram of fish. And this is an ongoing study, and we still have not done the challenge with the wild type, but we have some interesting results. That's why we want to share it with you guys. These are the 7 isolates that I used for this experiment, and looking at the results, we attained the mortality of 90 to 100% in this trial. And based on this analysis, we can see that 3 isolates out of 7 showed significantly lower mortality compared to their wild type counterparts, indicating attenuation. So we see a 2 to 3-fold reductions in the mortality in the RIF mutant tank compared to their wild types. So this trial is ongoing and protective assessments are to be done. So what are the conclusions that we can confer from this study? So we can say that all the wild types isolates are protective against E. piscicida S11-285, and also this indicates a potential for vaccine development. In the previous research, there has been evidence of cross-protective potential with E. ictaluri vaccines against multiple genetic groups. So we hypothesize that Idwasella piscicida also have a cross-protective potential across multiple genetic groups. And out of 17 tested mutants, we saw that 6 of them provided significant attenuation, and 3 have demonstrated some level of protection of around 75% relative percent survival against the wild type. So we need further efficacy testing. And I also have 2 more isolates that need to be tested for attenuation, and then we will go for the further research, which would be optimizing the immunizing dose for the protective candidates by targeting the relative percent survival values of more than 80%. From there, we will select the isolate affording the greatest attenuation and maximum protection, and then we will confirm the protection against all genetic lineages. So for the one final vaccine candidate which provided maximum protection, we will go through oral and immersion delivery models to check for the vaccination strategy, and also we will check cross protection against E. ictaluri, and all the immunological assessments will be done for that one excellent candidate. So with this, I would like to thank all the funding agency and USDA and University of California, Davis for the collaboration. I would like to thank my committee members and all the people from the Griffin Lab for all the help with the experiments. And with this, I'll take any questions.
Great presentation, Sujita. Folks, if you've got questions, feel free to put them into the chat or unmute yourself. All right, nothing out there. Getting compliments in the chat, though. Just check it out. Thank you very much.
Thank you, Dr. Files. Thank you, everyone.